Hello, everybody here, and thanks for attending the webinar. And I'm Carter Hall from GenScript. And today I'm going to share the recent progress on antibody engineering and GenScript, and especially the AI-based developability assessment of therapeutic antibody and its engineering. And the presentation contains following parts. And now let's move to the first part. And as we all know, therapeutic antibody is a very important class of therapeutics and grows fast. And as shown here, there were several antibody therapeutics got approved every year since 2010. And in past 2023, there were 16 antibody therapeutics got first approved. And until now, nearly 200 antibody therapeutics are available in total. The development of therapeutic antibody is always not that easy. Most part of the therapeutic antibody development are time-consuming with high cost and the traditional wet lab-based methods. And in some of the parts, the search scope is limited due to the library size, for example, in a humanization process. Well, fortunately, with the popular of application of AI in biology field, it gives us the chances for solve many problems in the development of the therapeutic antibody with AI in a more efficient way, more cheaper and more faster. And also, with the powerful computing ability of the computer, the search scope could be largely enlarged to break the limitations of the library size. And there exists about 82 AI-related biotech companies which pay attention on a protein or antibody drug discovery. And more than 60 of them are established in last 5 years. And so AI is more and more important, and the antibody therapeutic development is also no exception. So next, I'm going to introduce the AI applications in GenScript, the immunoinformatic platform. And here we list the general process of the development of a reagent or therapeutic antibody in GenScript, starting from the antigen design, go through with animal immunization, sequencing of the antibodies, and the developability assessment and antibody reformation production, and end with qualified products delivered to our customer. And in each part of the process, we have the corresponding AI bioinformatic tools applied. And today, we mainly focus on the developability assessment. Other tools will be also briefly introduced. And now let's go to the antigen selection and the preparation part. And as you know, antigen is always the first step of the antibody development. And GenScript have our own internally used antigen analysis packages. And here I briefly show you the epitope prediction tools, which is mainly for efficiently animal immunization to get the animal-sourced antibodies. We regard the epitope prediction problem as a classification problem to identify whether the given peptide is the epitope or not. And we plot the evaluation indicators for measuring the performance of the classification model in the figure. For positive and for true positive and true negative, the higher the better, and for false positive and false negative, the lower the better. And the performance of GenScript epitope prediction tool is plotted in blue, and we can see that our tool performs better than the comparison method. The epitope, which is published in 2021. And so with the epitope analyzed, and next step is to express the antigen. And actually, no matter what kind of the protein to be expressed, for example, the peptide, small protein, and antibody, the DNA sequence is essential for determining the expression level of the target protein. For example, the GC contents would obviously affect the expression and they should be optimized. And in our newest AI-based codon optimization method, Zhuque performs excellent in optimizing the codon to increase the expression level. And we take the previously codon optimization method, GenSmart, to make the comparison. And the above two figures show that Zhuque significantly improves the performance, no matter in human, mouse, or rabbit. And below list the advantages of Zhuque codon optimization method, especially the stations. Our codon optimization tool has been cited by more than 80,000 works in their publications. Animal immunization, ELISA testing, NGS sequencing, and the are followed with the antigen get prepared. And we also have some internal tools for analyzing the sequencing results for making sure that the sequences are correct. And with those sequenced antibodies at hand, we're going to do the developability assessment, including the basic sequence annotations, PTM aggregation and degradation sites analysis, and also immunogenicity, viscosity risk assessment, and also with the clearance rate and MHC-based immunogenicity, and so on. And so next one. Here is the antibody chain domain annotation tool for letting the customer know basic informations of antibodies on the sequence level. The system will separate the full chain into several parts, including the single peptide, tag, linker, V region, C region, and fusion protein, and marked with them with different colors. We also evaluate the quality of the CDRs and FRs. If the specific region is not satisfied with the evaluation criteria, bad flag will be marked. And other information like species, chain isotype, mutations are also provided in its analysis report. And GenScript also provides the bispecific antibody-related services. There are more than 1,000 kinds of bispecific antibody structures in our knowledge database. The structure of a given bispecific antibody can be smartly identified with the SAB structure annotation system. Each sequence will be split into several parts, and the chain ratio with the reference structures are also reported. And well, if the query bispecific antibody is not identified in our database, the most similar one and its corresponding information would be reported like here. So now our next in-house program for developability assessment are aggregation, degradation, and PTM sites prediction programs. And here for the aggregation, we compute the spatial aggregation prong score for each residue and identify whether the residue is potential aggregation sites or not. The upper right picture, it is a modeled 3D structures in mesh mode with SAP scores colored. The higher SAP score is, the darker the red color is, and the lower the SAP score is, the darker the blue score is. So as shown in the picture, there are the four regions seems to be potential aggregation sites regions, like here. And in this side, the figure shows the potential aggregation sites in this way. And we can say that those four regions would be identified as the potential aggregation sites. And so next one is the PTM. Oh, no, sorry, not PTM. It's degradation. We mainly consider asparagine denudation and methionine oxidation. With only those few features used, like here, we can achieve the prediction accuracy equals to 94%, 94%, and the AUC score equals to 0.96, which means a high prediction accuracy. And next one for PTM, we use the pre-trained protein BERT models as a base model with the use of antibody-specified PTM datasets for fine-tuning to get the different PTM prediction models for each considered PTM type. And here we list the prediction AUC score for those kind of PTMs. And then the test results show that the fine-tuned models achieve over 0.9 in terms of the AUC score, which also means a high prediction accuracy. And next one, I will introduce the viscosity and immunogenicity prediction. Here, if we could got those kind of the viscosity and immunogenicity information of the candidate therapeutic antibodies once the sequence has been designed. Yes, so, and then we can make the corresponding decisions for choosing which candidates to be produced and validated. Thus both of the time and money could be saved, right? And even we can make corresponding reformations of those candidates to treat off the developability indicators. And definitely, we have developed our own method for predicting the viscosity and immunogenicity. So here we show the pipeline for viscosity prediction. It is a large language model-based machine learning method. So start with only the antibody heavy and light chain as input, and we first extract the protein T5 embeddings as the representation feature of the sequences. And then the feature is compressed into low dimensions with the kernel PCA method, and further input to the random forest classifier to predict the viscosity value. And as the concentration at 150 milligram per milliliter, the viscosity value above and below 30 CP would be classified as high and low, respectively. So this work was published on the European Journal of Pharmaceutical Sciences in 2024. And this slide shows the performance comparison between several published sorter methods and our proposed method on the AB40 dataset. As listed in the table and shown in the figures, our method performs best, yes. And on another test dataset, AB21, our proposed method is comparable with the method proposed by Pfizer. And here, this one is by Pfizer, and this one is our GenScript prediction method. And we also take our method compared to the latest published viscosity prediction method from the University of Michigan on several existing antibody therapeutics as listed in the table. And then the test results show that our method predicts 100% correct, while the UM method just achieves accuracy that is 70%, yes. And similar to the viscosity prediction method, and our proposed immunogenicity prediction method also take a pre-trained large language model as the feature extractor, and with the use of autogluon as the classification model trainer for getting the immunogenicity prediction model. And this work has also been published in early of 2024, and both papers are available online, and you can get it for read for more details. Yeah, and here shows the prediction results in comparison to other methods. We take only one previous existing method, which predicts the 88% as the immunogenicity indicator for comparison, which was published in 2020, yes. And in terms of both AUC and PRC, our proposed method performs better. And as all of the currently implemented developability assessment indicators, our immunoinformatics service platform has been introduced. And so here I bring a good news to our customers or potential customers. All of those developability assessment prediction tools, they can be obtained from our TurboCho Smart Code online ordering system. It has already been released. And if you have interest in knowing the developability indicators of your design antibody, and you can then try it for free. And with those developability indicators at hand, our question for you may want to ask is, could you suggest me how to do the modification? So next, I'm going to introduce some antibody engineering services at GenScript. The first one is the monoclonal antibody humanization service. There are mainly 3 kinds of humanization strategies on the market. The CDR grafting, chimeric antibody, and the fully humanization. The fully humanization method always expensive and may encounter the patent restrictions. And while the chimeric antibody has considered considerably high HAMA risk, and so the CDR grafting method is more popular with low cost and fast delivery. So we have 2 ways for implementing the humanization: the traditional bioinformatics-based rational design and the AI-based design. Those methods are all on the basis of CDR grafting and in our developed humanist evaluation models. And this slide shows 2 rabbit monoclonal antibodies humanization cases. We show the BLI binding affinity test on rabbit human chimeric antibody and the humanized antibody. The test results show that in our humanized antibody do not lose the binding affinity, and the affinity is even higher than the chimeric antibody. And besides the humanization, we GenScript also pay attention on the pet care. So we know many people that take dogs or cats as pets with them. So pet health is also very important to them. And there also exists the needs for animal antibody engineering. And so we have also developed the corresponding animal-related services, such as the monoclonal antibody canonization and the felinization. And here we show 2 cases for we did before for rabbit monoclonal antibodies, canonization and the felinization. The figures are SPR binding affinity testing of the chimeric antibody and the canonized and felinized antibodies. From the values, we can see that the binding affinity of the canonized and the felinized antibodies have a slight increase, which means the successfulness of our canonization and the felinization procedure. And so we have summarized the heterolization methods as a scientific paper, and it has been published. And we can see the snapshot of the online server, and once the and it has been now can be accessed. And if you have interest, and you can type the website and have a try. And so another important aspect of the antibody engineering is the binding affinity maturation. And here we just list a case for doing the binding affinity maturation, which shows that compared to the wild type antibody, most of our modified antibodies have an increase of the binding affinity, while the increase is limited. We are still working on this with the recent popular large language model, and the newly developed method is now on wet lab testing. And except all of those LM-based AI-based antibody binding affinity maturation, the generative AI multi-species humanization and multi-model evolution-based antibody therapeutic developability reformation are also coming to see you guys. Well, last but not least, so I'd like to introduce the TurboCHO antibody expression platform. Currently, we only take 5 business days for high throughput expression and 10 business days for gram level expression, which is significantly faster than other providers, taking almost 6 to 8 weeks. And in our platform has received great feedback for its high yield, and we can even achieve a gram level production for transient expression. Meanwhile, our prices are very competitive, and for more details from our TurboCHO, please feel free to reach out to our GenScript sales team. So the next one I want to say is the Q&A part. Do you guys have any questions? Thanks for all your attention. Thanks.